Hello everyone, welcome to this Lessons Learned video. Today we're going to talk about glial cells. So you might wonder what glial cells are. In the nervous system, the cell type that you're probably most familiar with is a neuron. So a neuron looks something like this. You've probably seen the shape of this before. You have a little axon, cell body, then coming out of this we'll have certain, dendrites, and I apologize for not being able to draw dendrites very elegantly, but these are branched little extensions that will receive signals from other neurons, right? So this is a neuron, but the majority of the cells in the nervous system are not going to be neurons. Right? They're actually going to be glial cells that are going to support and nourish neurons and make sure that they're functioning properly. But neurons do the main, communication and sending signals and doing all of that stuff. So glial cells are different than neurons. There are many glial cell types that you probably don't need to be familiar with for an introduction to neuroscience class. And if you're just looking for basics, this is what this video is for. So the first cell type we'll talk about is astrocytes. You might recognize that astrocytes, astro means stars, so they look they have these branches from the center, and they branch out to many different areas. It's kind of what they look like, and they have this little nucleus in the cell body. Their main job is to clean up brain debris and to hold neurons in place. They can also do other things. So you can have a synapse between two neurons, a little space where two neurons communicate. So this is one neuron and this is another neuron synapsing with each other. And then you can have a little astrocyte. So we'll draw the astrocyte in purple just to demonstrate that it's an astrocyte, right? An astrocyte also extending into this synaptic space, and what this neuron can do, this neuron can, let's say this is the presynaptic one. It can, let's say, send neurochemicals, in these vesicles on there to the postsynaptic neuron, which will then detect them through receptors. So these neurochemicals bind to these receptors. That's how a synapse generally works. And then once they bind to these receptors, right, ions are going to flow into the cell through the ion channel that is next to the receptor. Okay. So these ions and these neurotransmitters, their concentration around the synapse is very important, and it needs to be very well regulated. And one of the things that regulates them are astrocytes. These astrocytes can come in and change, ion concentration, and can also change neurochemical or neurotransmitter concentration, and can move it around and give it to different cells. So that's an important thing that astrocytes do. Okay. Now another important thing they do. Let me erase this little star drawing of the astrocyte. The blood-brain barrier looks something like this. You have these endothelial cells, which are the inner layer of the blood vessels. And these endothelial cells are sitting, so like this is the central nervous system, so this is neural tissue on this side, and then we have the inside of the blood vessel on this side. And I could have drawn them red so that it would be cooler, so let's do that. They're endothelial cells and they're next to blood. Right. So you got these endothelial cells that are sitting here, and each of them has a nucleus, and they're held together by these tight junctions that I'm exaggerating here. These are very tight junctions between them. The purpose of the tight junction between these endothelial cells is so that substances do not enter into the central nervous system that we don't want. And this is called the blood-brain barrier. Blood-brain barrier, BBB, right? So the blood-brain barrier is going to be formed by these tight junctions of astrocytes. So neurochemicals are not going to, or not neurochemicals, but chemicals that we don't want in the central nervous system are not going to get in here. Like bacteria wouldn't get in here. Molecules that would interfere with neural tissue, because neural tissue is very delicate. You don't want to disrupt the chemical environment in there. Astrocytes can come in, and let's say this is an astrocyte cell body, and it can actually send little projections onto these endothelial cell tight junctions and can release certain chemicals. So it can release chemicals on there that either make the tight junctions more permeable or less permeable. So what these astrocytes can do is change the permeability of the blood-brain barrier. They can either make it allow more substances in, which makes it more permeable, or allow less substances in, which makes it less permeable. So these astrocytes can modulate the permeability of the blood-brain barrier. So those are astrocytes. That's generally what they do. Next, and by the way, astrocytes are the most numerous type of glial cell. Next, we have microglia. They look similar to astrocytes. They, when they're not activated, they look kind of sit in place, and they have these little branched extensions. So let's attempt to draw that. So they look kind of like this. And then when they are activated, they lose those branches and they become a cell like this. Now, microglia are the major immune cells of the brain. They would protect the brain from invading microorganisms. And they're also important for cleaning up debris. They're phagocytes, meaning that they engulf other things. So if there's a microorganism that comes in, right, this activated microglial cell would come in, and let's say this is a little bacteria that just invaded us. I want to make it look like sperm. Let's just say it has little feathery extensions on it. It would come in and engulf the bacterial cell. Right, so, or let's say we have a neuron, and let's say the neuron is, let's draw a bipolar neuron, right? So this is a neuron with little, that's not an dendrite, sorry. It has these dendrites, and it has a little axon on the other side. Let's say there's a little damage on it. So let's say it kind of, say it's broken, right? There's a bit of damage on the dendrites. If any cell shows any sign of weakness or damage, these microglia are going to come in and also engulf it. They're going to come and just destroy it, right? Going to eat it and not allow it to damage other areas. So they're important for cleaning up debris, and importantly cellular debris and damaged cells. So in that sense, they're going to be important for inflammatory reactions. Whenever you have an inflammatory reaction within the central nervous system, it's going to be microglia carrying that out. Microglia actually don't originate from the central nervous system. In development, they are derived from bone marrow. They're bone marrow-derived monocytes, and they come into the central nervous system during development, and then they stay there and don't leave. And they just wreck everything that they don't like seeing. So that can either be a damaged cell, or it can be pathogens. Okay, so these are immune cells essentially. All right, so that is microglia. Next, we're going to talk about oligodendrocytes. These oligodendrocytes are going to be what myelinate axons in the central nervous system. So let's say we have a neuron, and I'm not going to draw the dendrite because I'm tired of drawing it bad. But let's say that on the other side there's a dendrite, right? Let's say that it sends this axon out and goes and synapses to some other cell. And let's, excuse me for one moment as I do this. There. It didn't work, and I think my app might crash. Anyway. Basically, these oligodendrocytes look like this. They come in, and let's say that we have multiple axons. So let's say we have this neuron. All right. We have this neuron, we have this neuron. And we have another neuron that's just like, it's over here. What's going to occur here is that a single oligodendrocyte is going to be able to come in and myelinate a section of multiple axons. So an oligodendrocyte can look like this. And it's going to come in and myelinate here. And going to come in and myelinate over here. And then it can also come in and myelinate over here. Now, this is going to happen in the central nervous system. The point of myelin, it's really a mostly fatty tissue. Excuse me. It's going to surround the axons. And if you look at a cross section, let's say that we have an axon. We have myelin around it. So the myelin is going to be in these concentric layers. They're going to just surround over and over and over the axon. And then they're going to fold on itself and go all the way back. Okay. The purpose of the myelin is to increase the resistance of the axon. Now, what that means is that we're going to not allow the current or the electrical signal as it's going down, the action potential is going down an axon. If there was no myelin, it would just leak out. But since there's increased resistance here, it's actually going to stay in and move down to the axon terminal. So it makes it easier for the signal to propagate. Also makes it the signal travel faster. Myelinated axons are a lot faster. So it's really an insulating thing, right? So these myelin are insulators, kind of like the insulators on a wire, right? Your phone charger isn't just a piece of copper, right? It actually has covering around it for insulation. Now, these oligodendrocytes are in the central nervous system. In the peripheral nervous system, you're going to have Schwann cells. And Schwann cells are similar. There's one key difference that your professor might stress here, and it's that if you have a Schwann cell, it's going to myelinate. These are myelinating peripheral nerves. So this is like a nerve that's outside your spinal cord, right? Something like that. These are going to myelinate. Every Schwann cell is going to make one of these little blocky segments. Okay, so a single Schwann cell can't myelinate several axons in this way, right? And indeed, in a single axon, both in the central and peripheral nervous systems, you're going to have this segmentation of myelin, and in between, you're going to have nodes of Ranvier. Okay, so multiple Schwann cells and multiple oligodendrocytes are going to be myelinating one single neuron, both in the peripheral and central nervous system, respectively, right? So that is what myelin is and how it works. And by the way, if you ever heard of the difference between white matter and gray matter difference in the brain, that's what this is, right? Gray matter is neuronal cell bodies, like right here, where it's not myelinated. Then once you see axons, right, large bundles of axons, that's going to be the glistening white myelin, right? It looks white when you look at brain sections. You can tell the difference that way. Right? Where you have gray matter, it's a large collection of cell bodies, or unmyelinated axons, but mostly cell bodies, and then white matter is these collections of axons that are mostly myelinated. Right? Not all axons are myelinated. So the last type of glial cell we're going to talk about, and these are again most usually talked about in introductory neuroscience class, are called ependymal cells. Ependymal cells. These are going to line the ventricles. So the ventricles are, it's hard to draw them, but I'll try my best. So we have kind of these are large structures in the brain, and they're actually really not structures. I guess they are mostly empty space. I shouldn't say empty space. They are actually a fluid-filled space in the brain. Okay, so fluid-filled spaces in the brain, and their main job, is cerebrospinal fluid, which you may have heard of, is going to be traveling through these, and then it goes down, goes into the spinal cord. And so whenever you're doing, you ever heard of a lumbar puncture? That's basically the fluid gets produced in the ventricles here, and this is kind of, if you were to draw the brain out, I can't draw all the gyri and all the details, but it's kind of, brain is, let's do it in a white, can't switch, there we go. Brain is kind of over here, and then it hits around right here. You can kind of tell where that is, and then here we have the spinal cord in the medulla. So here's the pons. And don't worry if you don't know all the neuroanatomy. You'll learn it eventually. But that's basically what the ventricular system looks like. So these ependymal cells are going to be lining the whole ventricular system. All these spaces on the inside are going to be lined with ependymal cells. Excuse me. So ependymal cells are going to secrete this cerebrospinal fluid that's going to be secreted, and they're going to secrete it, and then the cerebrospinal fluid is going to travel through these ventricles and go down and into the spinal cord. They maintain also the composition of CSF, and they generally keep the composition of CSF very constant. So like the concentration of substances in there are going to be very constant and the same, and they're making cerebrospinal fluid and secreting at around exactly the same rate all the time. Okay, so the rate of making CSF is independent of blood pressure and intracranial pressure. Because the composition of CSF, or cerebrospinal fluid, is usually going to be constant, you can check for pathology. You can do a lumbar puncture, and if the protein concentration is too high, that might tell us that there's viral meningitis, right? Because you have viral debris. But if the meningitis is bacterial, we might have low glucose concentration because the bacteria is eating the glucose. Now, these ependymal cells are also going to make the blood-CSF barrier. So the blood-CSF barrier means that we're going to allow CSF to go into the blood stream through what are called arachnoid villi. For an introductory neuroscience class, you might not worry about that. But for neuroanatomy, you might have to learn that. Arachnoid villi are one-way gateways from the what are called the sinuses. Right? So more specifically, the superior sagittal sinus. These are basically big veins that carry blood away from the brain, and it's kind of, if I can draw it out here, it'd be nice. Right, so it's going to be kind of, let's draw. There's this big vein that goes over here, and the cerebrospinal fluid is going to be in what's called subarachnoid space. So it's going to be kind of in this very outer layer space, right? Almost close to the skull. And then arachnoid villi are going to be these channels that basically allow CSF to go into the bloodstream. And this is deoxygenated blood, but they're not going to allow blood to go into the CSF. That's not allowed, all right? That's the blood-CSF barrier, and that's going to be maintained and created by ependymal cells. So that's why they're important, right? They allow the creation of CSF, which CSF again is going to, supply neural tissue, neurons, and is going to cushion the brain inside the skull so that when there's a lot of movement, the brain is kind of, its apparent weight is decreased so it doesn't crush into the skull, right? So that's basically the entire video. So that's all the important glial cells that you need to know about and some basics on them. So thank you for watching.